Thank you for having me. I'm very pleased to share our research and findings with the plant breeding community. I'm ATANDA Sikiru Adeniyi by name. The topic of my presentation is methodologies to improve the efficiency of genomic selection in CIMMTY maize breeding program. These are the outline of my presentation. My background, introduction to CIMMTY genomic selection pipeline, where we are and where we want to be, materials and method, research and discussion, and conclusion from our results. Once again, I'm Sikiru Adeniyi by name. I have my BA Greek and MSc plant breeding from the University of Ilorin, Nigeria, and the host institution for my PhD program is West Africa Center for Crop Improvement, University of Ghana, while I have my research conducted at International Maize and Wheat Improvement Center, CIMMTY, Mexico, in collaboration with Kelly Robbins' lab in Coney. The presentation is actually an excerpt of my PhD research work, though I'm presently a postdoc with the Global Maize, with the Global Wheat program in CIMMTY. So I'll begin my presentation with the breeder equation because it's a useful starting point, being the fundamental application of quantitative genetics to crop improvement. It has parameters that breeder can manipulate to accelerate genetic gain, and the parameters are genetic variation, selection intensity, HAR, which is selection accuracy, and early detection cycle time, while sigma G is genetic gain. So the overall objective of this presentation is how the maize breeding program is using genomic selection to manipulate the parameters of the breeder equation and accelerate genetic gain in the breeding program. This is an overview of the CIMMTY tropical maize breeding program where we're coming from rather. So the breeder make an inbred, the breeder make a cross between two inbred line A and B, and such crosses can be in the 100 to generate F1. The F1 is served to F2, and from the F2 DH line are derived. The derived DH line a test cross to test that from opposite heterotic group. And the test crosses are evaluated in stage 1 in 3 to 5 locations, and the superior lines are advanced to stage 2, which are further tested on more testers, and the test crosses are evaluated in 5 to 8 locations. The superior line from stage 2 advanced to stage 3 and also further tested in 4 to 5 testers, and the test crosses are evaluated in more than 15 locations. Lines with high SEA and GCA are used in hybrid combination, and the hybrid are evaluated in more than 50 locations, which are eventually released, and the superior hybrid are eventually released to the farmers. So despite the success in the maize breeding program over the years, there's always an opportunity for an improvement and optimization. So the drawback of the phenotypic selection is the fact that it is expensive and time-taking. For instance, the stage 1, where the breeder, which is the preliminary trial, where the breeder want to evaluate the new line for yield potential, which are thousands of lines that the breeder want to evaluate at this stage, which make it very expensive. At the same time, the breeder has to wait 3 to 5 months, depending on the maturity of the maize, before advancement decision can be made to stage 2. So what are the opportunities available to accelerate genetic gain in this maize breeding program? The double haploid technology, which is already in use, the chip genotyping platform, and the use of the genomic selection to manipulate the breeder, the parameters of the breeder equation and accelerate genetic gain. So where is CIMMTY actually implementing genomic selection, currently implementing genomic selection in the testing stage, is the stage one. But in the stage one, given the fact that the number of lines that the breeder want to evaluate at this stage are enormous, and as such, the breeder can reduce the number of individuals that need to be tested at advanced line, and advanced promising line directly to stage two using the GBV. So what is the current genomic selection strategy that is being used in the CIMMTY Maize breeding program? Is test half and predict half, which implies that each biparental population is split into two, half of which is planted on the feed as the training set, which is used to train a predictive model and predict the genomic and predict the genetic merit of phenotype individual. So selection is being made with a GBV at the same time with the blue. So this is the empirical evidence of the efficiency of genomic selection in the CIMMTY Maize breeding program. This has been published in 2019, where individuals that have been selected using the GBV and individuals that have been selected using the selection advanced to stage two and planted on the feed. And so the result that has been shown here give clear-cut evidence that there is no difference in the selection accuracy using the GBV and the phenotypic selection at the stage one in the CIMMTY Maize breeding program. So what is the implication of the strategy, this current strategy? So with this strategy, you can increase the selection intensity, there's increase in the selection accuracy. However, we are unable to reduce the selection cycle time because the breeder still has to phenotype a subset of the parental population to train a predictive model to predict the genetic merit of the unphenotype individuals. So they're still delaying in decision making, and also the cost of generating the phenotypic record, which is used as a training set to predict the genetic merit of the unphenotype half of the parental population. In order to maximize the efficiency of genomic selection in the CIMMTY Maize breeding program and improve the prediction accuracy, so we propose a stepwise scheme, which basically involves the use of historical data as a training set to predict the genetic merit of new or biparental population. However, because historical data is made up of individuals that are closely related to the new population and those that are distantly related. So we use genetic optimization criteria to select individuals from the historical data that are most informative of the new population to train a predictive model and generate the GBV. So individuals are advanced directly to stage two using only the GBV. And the scenario where the historical data can now be confidently used to predict the genetic value or merit of the new population. The breeder can still use the test type and predict type and the sparse testing. However, with the use of genetic optimization criteria to select individual that will be planted on the feed and individual that will be shelved. In one of our publications, which I'm not going to present here, so we find that the sparse testing actually has improved prediction accuracy compared to the test type and predict type, given the fact that all individuals have phenotypic data, though in the different location. And in the scenario also where we want to, in the scenario we also propose here is phenotyping a few individuals from the population, from the new population, and augment the full safe training with individual that's optimized from the historical data. And we also find that this also results in improved prediction accuracy. So we don't lose accuracy even if we phenotype a few subset of the new population. and augment the full SIP training with historical data, optimized historical data. So these are the genetic material used to test and validate our hypotheses are materials from the CIMMTY Eastern Africa Maize Breeding Program in 2017 and 2018, which consists of 849 and 1389 DH line derived from 13 and 45 hybrid population. So the population do not overlap across years, and the genotype using repeats and amplicon sequencing. In order to test the hypothesis, so we first use the multiple biparental population, which has 45 population units, highly diverse data sets, and this is the population structure of the data set in 2018. The first approach was to predict a population using the remaining 44 population as a trend set. So in this case, I'm presenting only one of the results, which is population 8 as a training with individual, which are 84. And the remaining 44 population, which is 1,305 individual, is used as a training set. So we use different optimization criteria to select individual that might be informative of the population that we want to predict. So we select 50 individual to 1,000 individual. So the results shown here shows that optimizing the training set results in improved prediction accuracy compared to using all the individual in the training set as in the individual and the historical data as a training set. So with the use of, because we have enough half-seed in the training set, in the training set, so we use the pedigree information to select individuals that have half-seed relationship to the new to the population of interest that we want to predict. And we also have the result that's shown that using that as a criteria, it's results in improved prediction accuracy compared to using all the individual and the training set, all the individual and the historical data as the training set. Having this information, we also determine which of the criteria consistently result in improved prediction accuracy, and we found that the CD mean and FAGR, which is the average genetic relationship between individual in the training population and individual in the testing population, are two genetic criteria that we identify to have a potential of subsetting individuals that are most closely related to the prediction population. And this is the genetic coverage of the optimized training set using these three genetic optimization criteria. So the TS is a testing set, the TS, which is the blue line, is testing set, and the green, which is individual that's identified by the genetic optimization criteria to be half-seed to the testing population, and the chocolate is individuals that don't have pedigree relationship, but have been selected to be informative of the breeding population. And the TRN are individuals that are not selected, which is the black, individuals that are not selected at all by the selection criteria. So given this information here, it shows that the FAGRM as individuals that are within the genetic space of the testing sets. Likewise, the CD mean, the part of the CD mean are still within outside the genetic space of that of the breeding of the testing population. However, ME has basically most of the individuals have been selected are outside the genetic space of the population, which shows that it is not efficient in in selecting individuals that are most informative of the testing set. Having this information, so in order to further test the efficiency of the two identified optimization criteria, so we combine the data set in 2017 and 2018 using the 2018 data set, which is highly diverse, to predict populations in 2017. So what we found is the same as we have in the previous data set, but with high confidence that the CD mean and the FAGR consistently results in improved prediction accuracy compared to using all the historical data as a training set. And this is the genetic coverage of the optimized training set using the CD mean and the FAGR, which is similar to what I've presented before. So the conclusion here is the breeder can use the historical data to predict the genetic merit of new population by optimizing the historical data set to calibrate the predictive model to predict the genetic merit of a new individual and advance directly to stage 2 without testing the new population in stage 1. And also from the result, we found that it is unlikely to have a single large TP with high predictive accuracy across the breeding population. And in addition, for population that require targeted phenotyping of the foci, criterion such as the CD mean should be employed to select individual for phenotyping. These are my supervisors and collaborators. I'm grateful to them, and thank you for the invitation. And